Industrialization to create employment.
Aha akakiiko ka boda ka Uganda National Council for Science and Technology obukabana kashijuma emerumere yone ekorwa hitendekere ryarwe bitete muri kiruhure kashanganguriya kahabucwe kasha tunabushata harigana konke ndwara ya COVID-19 hino kwebara abanise mukando gwe bagendereye kweza mu mwaka gw'2023
ngono bija kweza obwire bwacho obatiro mu ndagano aha hamwe na namamve abo kuba hati tuyabucweka ashatu ndabushata ari 10 kandi no gwire butabe turaganire kukoramo nabwe bwingana na ecweke kikozwe
amatendekere agagarukuza kumaraho sente zero kurenga omaro billion 350 ngashajikwa government ya Uganda kandi sente zimwe bakavyeguzo mu government ya China itendekere ryarwe bitetere shitamira hari hika zero kwikana n'itano konka habasa kwija kuba hecetengo cyo kuriyongera mu buhangu.
And the initial plan was actually to maximize the utilization of this land. It is a campus, it is a learning center. And the learning center has to be big enough to accommodate the learners.
Abanyarwanda barukurenga 15 no barije kubana bashongorwa omyoge tarukushana kuba kwera kongera hantasa yihanga eyo mukuru wihanga agize ngo neja kubemerera hari US billion dollars anani no mukwezi.
The establishment of a multi-billion business incubation center at Namamve has taken shape.
Prompter here or remote for your presentation, please focus it towards this side. The signal is on this side. It's not at the front where the monitors are. It's on this side. And then try to be as close to the microphone as possible so that even our online participants will be able to follow properly. Thank you again, and thank you virtual participants for being actively involved in this event. Without much ado, I wish to welcome the chair for this session. He is the director of Uganda Virus Research Institute and the Medical Research Council, Uganda Virus Research Institute Research Unit. He is an honorary professor of the London School of Tropical Medicine, and he's also a professor at Makerere University College of Health Sciences. He holds a doctorate in immunology and has played a critical role in HIV research in Uganda, especially in the understanding of the protective immune responses, HIV diversity, and resistance to ARV therapy. He has served on several committees, including the WHO HIV Vaccine Advisory Committee, the Global HIV Vaccine Advocacy Coalition Board, the CHAVI Immune Discovery Scientific Advisory Board. He has also chaired the African AIDS Vaccine Program, among others. And he has also served on the Uganda National Council for Science and Technology, Health Sciences, Specialized Technical Committees. So this is just a few of them. We could take all day talking about Professor Kalebo. You're most welcome, Chair.
Thank you very much. You have a slightly old CV. We shall update it. I'm actually a full professor, not an honorary professor of London School. And I no longer have the honorary professorship of Makerere. They never renewed it, I don't know why. But I'm a full professor at London School of Hygiene and Tropical Medicine, of Immunovirology. The rest is okay. Welcome, good morning everyone. And thank you, the organizers, for inviting me to chair this session. This is our third day of this annual National Biosafety Forum, and I'll be chairing this session number 6 with the title Emerging Biotechnology, Technologies in Human Health. We are going to have a number of presenters, and I know we are behind time by about 1 hour. So I'll request all our presenters to keep the time. And before the presenters start, I'll be giving a short introduction, a short bio of these presenters. The first presenter will be online is not physically with us. I hope he's connected. And I didn't have a full, more details, but I know that Dr. Roy Mogira is the CEO of the National Biosafety Authority of Kenya. And we are very pleased and privileged to have him give us the first presentation, which is current regulatory approaches for gene editing and synthetic biology. We shall have our timekeeper. Oh, he's not online, unfortunately. So we'll skip that and we'll go to the next presenter, who is a colleague from Uganda Virus Research Institute. Dr. Sheila Balinda is a molecular virologist working at UVRI and MRC UVRI in London School. She's involved with characterization of HIV-transmitted viruses in Uganda, and she's also one of the scientists who are leading one of the viral vector vaccines for COVID. So she's going to present to us the health applications, gene editing, and other emerging gene technologies in Uganda. I've known Sheila for many years, and I've seen her developing, and she's one of those upcoming molecular virologists, and we look forward to your presentation, Sheila. You have up to 20 minutes to give us this presentation. So, may I request all the other presenters also to come in front, so that they can come in front. So but before I took Sheila into HIV and human viruses, she was working on foot and mouth disease virus. So she has been this virus. Okay, Sheila, can you please come and give your presentation?
As we wait for the technical team to project my slides, oh, thank you, they are projected. I would like to thank the chair for the kind introduction. And then I would also like to thank the organizers for the opportunity to make this presentation to you all here today. And thank you for being present. So the title of my presentation is Health Applications of Genome Editing and Other Emerging Techniques in Uganda. However, the key examples I'll get into will be specifically the work we've been involved with at the Uganda Virus Research Institute and the MRC UVRI, London School of Hygiene and Tropical Medicine. So a brief outline of my presentation is that I will briefly talk about gene editing and then get into the specific vaccine research we've been involved with for HIV. I'll then also give some specific updates with regard to the COVID-19 vaccine development in Uganda, and then next steps. So as you're all aware from the presentations that we attended yesterday, genome editing technology alters the nature of an organism through adjusting some of its genes, through replacement, addition, or total removal. And it's quite applicable in various fields. Specifically for today's discussion, we will focus on the health front. For example, therapeutics. As you are aware, before the age of genetically engineered drugs, for example, the insulin, we had significant challenges. For example, several animals like sheep had to be killed to come up with a substantial amount to address the key problems on the globe, or specifically at that time in the US. However, with the emergency of the genetically engineered, that is the recombinant insulin, you see that globally, diabetes 1 is currently under control. There are so many examples. I'll not get into them, but I'll just briefly highlight key specifics in the various areas. With regard to diagnostics, we do have reporter genes such as luciferase and the green fluorescent protein. These, when attached to other genes, they could be used to investigate in vivo, which would be quite challenging, key diseases such as cancers. And this is quite critical for diagnostics. Additionally, we do have the CRISPR-Cas technology that is quite critical in diagnostics of infectious and non-infectious diseases, as well as more recently, it can make distinction between SARS-CoV-2 viruses. And then my presentation today will actually focus more on the role or the critical advantage of gene editing or genetic engineering with regard to vaccines. I will start with HIV vaccine research that we have been involved with at the Uganda Virus Research Institute. As you're all aware, 40 years down the road, we still don't have a vaccine. Development of a safe, effective, and scalable HIV-1 vaccine and cure strategies still remains an urgent need. Knowledge on the biological properties. By biological properties of HIV, I mean, do we understand the virus well enough to be able to intervene? So here we would need to understand its replication capacities. How does it, which co-receptors does it use? And most of the studies that have been ongoing have majorly been in the West and focused on the subtype B and Cs. However, here in Uganda, through funding from the International AIDS Vaccine Initiative, as well as collaborative activities, we were able to participate and characterize the viruses that are prevalent in Uganda. That is subtype A, D, and the recombinants. As you're all aware, 60 to 80% of the heterosexual transmissions are established by a single HIV variant. It turns out that an infected individual has a swarm of very divergent viruses. However, not all these viruses have the potential to cause successful infection in the naive partner. It's usually one or two that have the ability to cause us. So it's not quite easy to build this capacity. However, over the past 5 years, we are able to do this at the Uganda Virus Research Institute. And you may wonder, so what? The key role here is when you have pseudoviruses, you can then do the downstream experiments. For example, you can then determine that these envelopes, what receptors on the cell do they use? We know what subtype B and C are using because of the research that has been done in the Western world. Is it the same case in Uganda? So we were able to characterize our viruses here, and we know the co-receptors they're using, and this is quite informative for therapeutic interventions. How do they come in and block so that viral entry is prevented? Additionally, you're aware that we have these new broadly neutralizing antibodies that have been isolated and have the potential to neutralize several antigens, meaning several viruses of HIV, different viruses of HIV. We also have the opportunity to test our virus strains against these broadly neutralizing antibodies to see if they can be neutralized. And this will inform the manufacturers to see if Uganda actually needs a particular prototype or we have one standard. So we are contributing to research that is actually relevant in our environment. We have also, in addition to the envelope, which is a critical gene for viral entry, we have also been able to look at the GAG and the GAG pole. These are structural genes, but they may also be relevant in informing about properties or characteristics of the virus that are actually important for consideration during vaccine design, as well as disease progression. So we were able to amplify the GAG pole, and then using the NL4.3 backbone, we were able to insert our GAG and GAG pole into the NL4.3 backbone, and then we performed downstream characterization assays using cell lines, commercially available cell lines. Specifically, we focused on replication capacity of the GAG poles. How does this gene impact on replication? And how does it relate to disease progression? These are all critical in understanding which of the viruses may be more detrimental to an individual during disease progression of the HIV or AIDS. We have also moved further, advanced from specific gene target studies to full infectious molecular clones. Here it means you're looking at the entire genome, which is quite informative because then you get to compare your work, the full genome with the specific. Are there variants, and can you come up with certain hypotheses for why there are differences? So with the full genome, we specifically used a totally different strategy because it was a bit challenging to come up with a full infectious molecular clones. So here we use a strategy referred to as chemical synthesis, whereby you identify the genome of the virus you're working in, you share this genome with a commercial company that then chemically synthesizes the entire genome of HIV from the first nucleotide to the last. They send it back to you in 3 pieces because of biosecurity, and then when you receive it at the lab, you come up with primers, and then you amplify the different sections and cause overlap and end up with the circular, I'm sorry, the pointer is not working, the circular full plasmid of the HIV at the bottom. The importance of this is additionally, you can label with luciferase or GFP and then use this in viral inhibition assay. The overall goal here with your viral inhibition assays is to identify key epitopes of HIV that can then be included for vaccine design. And it's very relevant here because working with our viruses, then you're able to identify those key epitopes that are relevant to Ugandans based on our prevalent subtypes. This work is still ongoing. As you're all aware, we still don't have a vaccine, but we do know more. And hopefully we are almost there. And we have the capacity, I'm happy to report, we have the capacity to participate and have our data compared on the international scene and contribute to new immunogens under design. Yeah. I will now switch to COVID-19 vaccine design in Uganda. I'm happy to report that the UVRI and MRC have had an opportunity, and specifically the novel adeno COVID-19 vaccine is a collaboration between Uganda Virus Research Institute, MRC, UVRI, London School of Hygiene and Tropical Medicine, as well as Makerere University College of Veterinary Institute. The key objectives for the COVID vaccine study were to develop a Ugandan non-human primate adenovirus vector. Why would we be making a vector when they are commercially available vectors? One, as Brenda highlighted yesterday, it was quite challenging to access these adeno vectors outside existing patents. So this was another challenge. Secondly, there was the issue of pre-existing immunity for those that were readily available on the market. Ugandans, through previous studies, had been shown to have pre-existing immunity like to the AD5 that is already commercially available, meaning even before you make your vaccine, our population has the ability to neutralize the carrier or vector that is to help you deliver the vaccine in the body. So that was not an option for us. We then looked at our capacity and realized with the training we had received under HIV, we could embark on coming up with our own adenovector backbone that could belong to Uganda and is easily available. The second objective was then to generate SARS-CoV-2 vaccine using the strains that were prevalent in our environment here, and then capacity building for future epidemic responses. Before I get into the progress we have made, some people may think that COVID vaccines are not relevant anymore. However, if you look at that map, specifically you can see Africa is light green. That is based on June 21st, 2022. It's an indication that in Africa, only 24% of the population has received the first dose, and only 18% are fully vaccinated. Therefore, compared to the world, we are still not doing okay. And in Uganda specifically, we are at 34% vaccinated and only 23% fully vaccinated. And as you're all aware, these are viruses and they are changing and changing pretty fast. Recently, the genotypic report highlighted that we did have a recombinant between Omicron and the Delta. It seems harmless, but we cannot be sure how all this will progress. So it's always better to come up with ideas and ensure that the population is fully vaccinated. So to address the objectives I previously highlighted, we collected a total of 108 chimpanzee fecal samples from the sanctuary and the wild, and we got relevant approvals. We then conducted sequencing using next generation sequencing technology, and we were able to come up with a range of viruses. We focused on the samples that gave us a signal for adenoviruses, and there were about 9. From the 9 samples, we were able to use, again, next generation sequencing, specifically the Oxford nanopore, and we generated the entire genome of the adenoviruses. We compared our adenoviruses with those that are actually in the environment. Those highlighted in blue are our samples from Uganda, and as you can see, they are significantly different from what exists in the public database. So we picked 1 or 2 to begin with, because this means these are unique viruses, and the backbone we end up with is different and is patentable. So we then proceeded to the cloning strategy. The cloning strategy here, we procured a commercially available Pac19 vector. It's an open system. However, you need to customize it so that it's able to receive your adenoviruses here. So we amplified small sections of the LF1, LF2, LF3, and inserted them into the vector. That way, during the homologous recombination approaches, where we are inserting the full genome of adenovirus, we are then able to insert it into this commercially procured Pac vector, the carrier of the whole genome, into one set. I'm sorry, the pointer is not working. No, yeah, it's fine. So we now generated at the bottom on the right-hand side, we have our adenovector down there at the bottom that has the E1 gene deleted. This is the one responsible for replication. The overall goal here is to ensure when we make our vaccine, we don't want it to continue replicating in the body. So you knock out this gene and you're sure replication will not happen. We then proceeded and amplified the spike gene from the COVID strains, and then again using homologous recombination, we inserted it in the vector. And I'm happy to report we have several vaccine candidates. 1, 2, and 3 are actually ours inserted in our viral vector generated from our adenoviruses. And then we got the CAV2, which is already characterized, and we also inserted, so this is like a positive control that will help us benchmark what we have made here. I'm happy to report for the third objective, we have also included 2 PhD fellows, Christian Dekize, they are all registered at Makerere University, and Dennis Omara. So Christian will be involved with characterization of the chimpanzee adenoviruses and determining the level of their neutralizing antibodies in the Ugandan population. That is pre-existing immunity of our backbone, how relevant is it? And then Dennis will look at immunogenicity and efficacy of our adenovirus vectored vaccine. I'm happy to report that we are already in the preclinical. We've set up the groups, and we are now beginning to test our products in mice. Our next steps will be to complete the preclinical studies and get this information. If it works well, we'll then plan to progress to the clinical studies. And then it's quite critical, at least for the backbone generated, if we don't have pre-existing immunity as Ugandans, to proceed to patenting this. It's an open system and can be used beyond COVID-19 vaccine in animal health and other priority diseases. For HIV vaccine, as I highlighted, we still are doing the basic science research. At the moment, we are looking into viral diversity of the contemporary viruses and effects on recombination for vaccine design, specifically the broadly neutralizing antibodies. Thank you all for listening, and I would like to thank the funders of this study, IAVI, USAID, as well as the Uganda government for the COVID-19 funding we've received. It's 100% funded by the government of Uganda. Thank you all for listening.
Thank you very much, Sheila, for that interesting and exciting presentation. Next, we have the next speaker, a colleague and a friend who has always been following me. Followed me at the medical school, followed me at the Zambia hospital, but now she's the executive director of the Joint Clinical Research Center. Dr. Sise Chitio Mutuluza has MBCHB, a master's and a PhD, and a fellow of the Uganda National Academy of Sciences. She's a medical doctor, public health specialist, seasoned researcher with over 28 years experience in conducting and coordinating AIDS research and care for HIV patients. She has a master's degree from Johns Hopkins University, School of Public Health, USA, and a PhD from the University of Amsterdam, Netherlands. She's currently the executive director of the Joint Clinical Research Center. She's truncated, but a long history and a good, very, very strong CV, has published quite widely, as I've mentioned, and she's now going to present to us on the opportunities in gene therapy in Uganda. Dr. Sise.
Thank you, full professor, and my senior and mentor. Today, I will talk about opportunities for gene therapy in Uganda. And Sheila has set the stage. I will not explain gene therapy in my presentation. I only have 20 minutes. But really, the basis is gene editing, to replace, to silence, or to introduce genes for purposes of cure of different diseases. So gene therapy is the next generation of medicine. So that is one opportunity already there. And while the rest of the world is moving ahead, if we don't join them, that will be our problem. So the opportunity is there, so we take it. And cure prospects using gene therapy presents a very promising new paradigm for management of HIV and sickle cell disease. And I focus on these, but gene therapy can be used for many other diseases. First of all, basically it is a one-time treatment to cure incurable diseases. It is potent, safe, and provides quantity and quality of life where it is applied. So the gene therapy, gene and cell therapy is booming around the world. You can see on the left-hand side, those are the numbers of studies that are taking place in the world. And that graph shows the number of applications that have been made to FDA in the US. It is projected that about 10 to 20 new cell and gene therapy products will be coming onto the market by 2025. But also, importantly, there is a lot of money being spent, but there is also a lot of money to harness. So as a country, we should really take advantage of this. This is a sample of therapies that have been approved by FDA in the US. You can look at the price tags one time, but look at the price tags, and all of these are given once, and then you get cured of your disease. So this slide here shows the red or the crosses, the countries in the world where gene therapy is taking place. And on the right-hand side, you see the pie chart. It shows USA is the biggest contributor here. When you look at sub-Saharan Africa and Uganda, I don't know if you can see Uganda there, there is no gene therapy taking place in our region. So we've been excluded. So should we continue to lament? What should we do? This is a survey on the clinicaltrials.gov, and it shows the ongoing gene therapy trials. This was accessed latest November 2021. You can see Africa, one gene therapy trial. This trial has taken place, it has actually ended in South Africa, and this is for hemophilia. So you can see where we are. And this, I think, alerts us on what we should be doing as a country. I'll now focus on two diseases. I said gene therapy can be done and used to cure many different diseases, but I'll focus on two, HIV and sickle cell disease. As you can see in the blue circles and the numbers above, sub-Saharan Africa has the biggest problem of these two diseases in the whole world. For HIV, we have about 67% of 38 million patients of HIV in the world. So while we have the least technology, we have the biggest problem of diseases where trends are moving. So for HIV in particular, we have had 34 years of antiretroviral therapy, but HIV remains the leading cause of death. And still many people can't access antiretroviral drugs. We are seeing donor fatigue because our governments are not the ones putting in money to treat HIV. So if we had a cure, wouldn't it be wonderful to treat everybody who has HIV? And one of the products that has been licensed is called Zinteglo. Zinteglo is used to cure beta thalassemia. Beta thalassemia is closely related to sickle cell disease. They are both hereditary diseases. And below you can see the price tag. If you don't have 1.8 million, you don't get this product. So this slide shows the progress that has been made elsewhere in the world with regards to these two diseases. At least 7 clinical trials have been opened between 2014 and 2021, and 36 clinical trials have used some bit of gene therapy to treat HIV. And how many of these have been done in sub-Saharan Africa? 0. So it goes without saying that in Uganda, no gene therapy trial has been done. It is also 0. But when we hear news of cure, it sparks hope. Many of you may have heard about the Berlin patient, the first one, Timothy. Now there is a London patient. There is Victoria Gray, who has been cured from sickle cell disease, but there are others, and these have been cured through participating in clinical trials. You have recently heard of the New York patient, and the numbers are growing. So now we want to hear the Uganda patient. So why is gene therapy important for Uganda? When you look at HIV, we have a high prevalence. We have about 1.4 million people in Uganda with HIV disease. And we have antiretroviral drugs, but people get tired of taking these drugs. And this has led to development of resistance to these drugs, which is another problem. Again, as I mentioned, there is fatigue. There is donor fatigue. Are these donors going to continue to provide drugs for HIV for Uganda for 30 more years? We don't know. Nobody knows. When you look at sickle cell disease, Uganda is the 5th highest prevalence of sickle cell disease in sub-Saharan Africa. We know that there are drugs like hydroxyurea, but very few people can actually access this. So I think our goal, given all these challenges, but they also present opportunities, is to move ahead and embrace gene therapy and cures for these two diseases as a starting point. These are some of the opportunities for gene therapy in Uganda, and I can't read that text, so I will face this way. There is a promising cure, and there is a lot of research going on. I've already shown you. We could be in Uganda a regional center for care and treatment, actually for cure, and that is what we are aiming for. And when we develop infrastructure here and do work here, we can get IP, but we can also generate money as a country. We know, I've already shown, it goes without saying, if I cure you of HIV, the quality and quantity of life will increase. And then we have an opportunity now to transfer technology. We don't have all the technology we need, but we can do the transfer. This is a slide that shows the roadmap to getting antiretroviral drugs in Uganda. We rolled out antiretroviral drugs for treatment in 2004. But the first drugs were available much earlier, and treatment was happening elsewhere. So, with regards to gene therapy, should we wait for 17 years? Or should we get proactively engaged so that we can shorten this timeline? So what now? What should we be doing? So gene therapy, because of these issues and non-involvement, we have established what we call the Global Gene Therapy Initiative Working Group, which was formed in 2020. And the goal is to enable access and implementation of gene therapy research in low and middle income countries. And specifically, we have selected 2 countries, the Uganda at the Joint Clinical Research Center, and then India. And the goal is to have the first trials for these 2 diseases by 2024. And the founders of this Global Gene Therapy Initiative, which we call GGTI, is that doctor who is based at the JCRC, and Professor Jennifer Adair, who is based at the Fred Hutch in the US. And we have built a network of, this slide is very slow. We have built a network of about 70 experts in various fields around the world, and we meet every week to discuss and see what the way forward is. We have community and patients at the center of the discussions, and all these, the experts, these are different groups, and these are the different organizations. where the people in GGTI come from. So how are we doing this? We have selected 4 pillars, and we are working on these 4 pillars concurrently to make sure that gene therapy comes and we meet our goal by 2024. This is one of the capacities which is very key to collect the first cells that you have to modify, genetically modify to produce the product, and we already have that. This product was developed by an NGO. This is the product that we are going to use at the JCRC for clinical evaluation. And the other product for sickle cell disease was produced at the NIH. It has been licensed to Caring Cross. And I talk about these products and evaluation here because they are very reasonable costs, so we can afford them. We wouldn't be evaluating anything that we are not going to be able to access. We've already gotten some seed funding, and we are doing capacity building. Lois is one of the trainees. She's at Fred Hutch and learning how to manufacture gene therapy. But we have others that have clinical placements and fellowships in the US. Manufacturing is the most costly aspect of gene therapy, and we are not going with the gold standard because we cannot sustain it. So we are already designing new platforms and also working on a mobile GMP facility within a container so that we can look at sustainability and manufacturing in country. Community is very critical and is part of this program because we have to engage and train the community. There are a lot of myths. We did the first vaccine trial for HIV in 1997, 99, and on the very day when we had to vaccinate, a politician went on radio and said we are guinea pigs and some participants didn't turn up. So we have to educate the community about what we are doing so that they embrace what we are planning. And regulatory, now this is very key, and we don't have guidelines and necessary regulations because this is the first time we are doing this. We've been talking to National Council of Science, we've been talking to the Biosafety Committee, we've talked to National Drug Authority, we've talked to the President, and WHO, they also has been engaged, both local and in Geneva. And WHO has just completed consultative process to develop guidelines on gene editing, especially for low and middle income countries. And these documents, they've already assigned a consultant to help us to incorporate gene therapy for health into the JERA bill. And in conclusion, gene editing tools are fast evolving and their applications are vast. So we need to domesticate governance and regulatory frameworks for research, clinical care, community engagement, infrastructure, and capacity building. Uganda will serve as a test case. That is the opportunity for implementing of the WHO governance framework for human genome editing, and JCRC in collaboration with experts within the GGTI and with support from government institutions, regulatory authorities, ethics, and biosafety committee, is pioneering the development of gene therapy products in country to address public health needs. And I have a short video. Can someone help me to play this?
My name is Brenda. I am a youth living with sickle cell disease. I am always in and out of hospital. But one thing I'm sure of is that there is someone always out there searching for a cure for HIV and sickle cell disease. And one of them is GGTI.
Gene therapy initiative is for the research to cure HIV and sickle cells, allowing diversity of stakeholders others to bring gene therapies to LMIC. Global Gene Therapy Initiative is for the research to cure HIV and sickle cells, allowing diversity of stakeholders to bring gene therapies to LMIC. We're gonna fly, fly high with GGTI. We hope to cruise safe over the mountain of pain, promoting access to where majority lives. We're gonna fly high with GGTI. I am a person living with HIV. He's a person living with sickle cell. Adherence and access to medication is a daily challenge we face in our lives. That's why we love the work you do. GGTI. We treasure the time you spend to save our lives. That's why we love the work you do. GGTI. We treasure the time you spend to save our lives. We gonna fly, fly high with GGTI. We hope to cruise safe over the mountain of pain, promoting access to where majority lives. We gonna fly high with GGTI.
Majority of the people with HIV and sickle cell reside in low and middle income countries. The Global Gene Therapy Initiative Working Group will initially focus on establishing a sustainable pathway to implement cell and gene therapy at the Joint Clinical Research Center in Uganda and CMC Center in India. It's time to train, translate, and initiate clinical trials for sickle cell disease and HIV in Africa and Asia.
Global Gene Therapy Initiative is for the research to cure HIV and sickle cells, allowing diversity of stakeholders to bring gene therapies to LMIC. We're gonna fly, fly high with GGTI. We hope to cruise safe over the mountain of pain, promoting access to where majority lives. We're gonna fly high with GGTI.
Doctor, my question to you is, what is your message to us as community regarding the GGTI in our country?
GGTI has identified pillars which are challenges and gaps in the delivery of gene therapy in low and middle income countries. And it is working concurrently on these pillars to fast track gene therapy delivery and access. However, this is a long journey, and in the meantime, we strongly advocate for care for HIV and sickle cell with available best treatment so that these patients are alive and available when gene therapy cure comes.
Thank you. Doctor, what kind of support do you need from us as community of people living with HIV, but also those that have sickle cell?
The support we need beyond even community is expertise in gene therapy, which we largely have in GGTI. But we need to build local capacity to lead the way. We need funding, absolutely. We are talking to the government of Uganda. We've gotten seed funding from Gates Foundation, and we are also talking to development partners. And then, very importantly, we need community support in advocacy, also for funding, and also information to the rest of the community, demystifying gene therapy, and then also participation in the trials when they are here. Thank you very much. And we are still lobbying government to help us with money, with gene therapy.
Thank you very much. As a colleague, I gave us some extra minutes. So thank you for that. So next, we are going to have now another presenter, and that will be Dr. Joseph Encordio, who is going to present to us on biosecurity interventions for national development. He is the National Biosafety Officer, Minister of Health, and currently coordinates the Biosafety Biosecurity program in Uganda. He holds a Master of Science in Microbiology and MPH from Mbarara University of Science and Technology. He has more than 10 years experience in the safe and secure handling of infectious biological threats in the country. He has received his formal training in biosafety, biosecurity from the National Defense University, USA, and the Sandia National Laboratories, New Mexico. He is certified by Risk Management and Biosecurity by the International Federation of Biosafety Association of Canada. He is IATA certified, and he has also vast experience in outbreak investigations and response to emerging and re-emerging epidemics and other public health services. So it's my pleasure to welcome Joseph Encordio to give his presentation.
Yeah, thank you, Professor. Joseph Encordio, doctor in making. I'll take on the journey. Good morning, ladies and gentlemen. All protocol observed. Like he has mentioned, my biography, I will not repeat it, but I'm glad to be here to share with you what the National Coordination Office on Biosafety Biosecurity Program in the country has so far managed to support our different stakeholders. I'll take you back to the year 2015 when we were reminded of the IHR requirements that state that state parties develop, strengthen, and maintain their capacities to respond promptly and effectively to public health threats. And this came as a call when we had the Ebola outbreak in West Africa, where we had most of our continent citizens being wiped out, and it gave birth to the global health security agenda, which one of the call was to make sure we have our laboratory capacity developed, and under the laboratory capacity, we had the component of biosafety, biosecurity being measured for the good practices. Prior to the IHR, we had to launch as a country the support for implementation of the global health security agenda, and it became very clear that we needed to strengthen our biosafety, biosecurity action package, which was under the laboratory capacity building. I am bringing this very vividly because all of us are talking about biotechnology, biosafety, biosecurity, and we have labs that are dealing or handling these very interesting technologies that we are trying to buy into our country to be able to embrace the call for a safe and secure work environment. And our country like I mentioned, when we embraced the global health security agenda, we had to do a baseline assessment. And what I'm projecting there is what our status was. And it was measuring two items in regards to biosafety, biosecurity. We had to take on the whole of government biosafety, biosecurity systems. And we found out that much as some MDAs or government agencies were practicing biosafety, biosecurity, but it was under, the score was limited capacity because some other ministries were actually in the slumberland. Then when it came to assessment on biosafety, biosecurity, training and practices, by that year, 2015, we had no capacity. And that was it. So we realized that I want you to measure, start measuring. After 7 years, what has happened? I don't want us to miss that slide. After the assessment, all of us know that all the technologies we are manipulating for the betterment of our country, in case anything went wrong, our home becomes Ministry of Health because we run there to seek for rescue. So in response to the assessment, Ministry of Health established a well-structured biorisk management program and created also the National Biosafety Biosecurity Coordination Office and put it under the Department of National Health, Laboratory and Diagnostic Services. And also we had to work with the different MDAs. As mentioned or as projected, we had Minister of Agriculture, Minister of Defense, Minister of Water, Wildlife Authority, and the primary focus was on making sure the biosafety, biosecurity competences and compliances are achieved. Allow me share with you what transpired since then. We managed to have a national training curriculum and program for biosafety, biosecurity, together with our biosafety manual. And initially, this training focused on the laboratory workforce, since everybody thought biosafety, biosecurity should be kept in the lab. But in 2016, we had to expand the scope and reviewed our curriculum to embrace and created a standardized curriculum which has three tracks, track 1, track 2, and track 3. Track 1, which is the basic track, we managed to train our support staff, the nurses, the medical doctors, and our maintenance engineers in the different institutions. Track 2 remained for our technical staff who are doing the real manipulation. Track 3 targeted our top and middle level managers, where most of us lie. And I would pose a question, how many of us have taken on a biosafety, biosecurity training at a management level, so that you get to know what you need, what is your role, what do you play with to make sure biosafety, biosecurity is up to date in your different institutions. The glaring gap in implementation remained due to the limited knowledge and buy-in from the top management. And this is where decisions are taken. This is where allocation of resources for biosafety, biosecurity takes place. So far, the National Coordination Office has managed to have 67 certified professionals by IFBA. IFBA is the International Federation for Biosafety Association. We have a number of them stipulated. We have those who have been taken through biosafety, biosecurity. Then we have the people who have been certified under biosafety cabinets, and then waste management. We also have the pool of national trainers, and these are helping us to cascade biosafety, biosecurity training in the different institutions under the One Health concept. In 2017, we had to do business differently. After training people for those 2 years, we realized that training alone could not produce results. So we had to come up with a biosafety checklist using the ISO standard 15190, which by then was the only standard which was supporting safety. And when we did an audit, the score, as per that time, it was 33%. And when we conducted another audit in 2018, after a year, we got 45%. This year, with support from Dr. Cisse of GCRC, we managed to conduct another audit, and I want you to bear in mind that with her support, we have another result to share with you in this forum as we go on. Yes. Without wasting time, you can see that in 2017, we were on 33. 2018, we went to 45, but we had a decline in 2022 by 12%. And the reason is very simple. We are now doing things differently. We've embraced international standards. We have the ISO 15190 and the ISO 35001 that support biosafety, biosecurity management for all institutions and laboratories. So I call upon the members here to pick interest in this, because we are all doing a lot of work in biotechnology, and we are doing them in the lab. But what is the status of those labs in regards to the ISO standards that we are supposed to embrace? I want to share with you another assessment. After the 2015 assessment by Global Health Security Agenda checklist, we had another assessment recently, that is last year, and this is what is the picture. I hope you kept in mind the score from the assessment of 2015, and this is what is happening in 2021. And this is published. We are still having developed capacity, but still we are scoring 3. 3 means we've not moved as per the international standard. And also in terms of biosafety, biosecurity, it shows that we've developed capacity. But the level of capacity does not allow us to move to higher scores, because some of our MDAs have not yet taken on or done their roles as the way they're supposed to do them. So, and we can't just score a Ministry of Health facilities alone, because this is a national scorecard that targets a One Health approach. Yes, after that score, we've now decided to make sure we review our biosafety manual and the national curriculum to cater for what would have been missed in the previous one, in the previous versions. And also, we've gone ahead to create the national mentorship toolkit so that we create biosafety, biosecurity mentors to support our facilities so that we can improve the projected score. We've also gone ahead to develop the national healthcare waste management guidelines, which used not to be there because we already now have a policy, and under One Health, it will be reviewed to embrace the different MDS to make sure biosafety, biosecurity is part of the different sections. Now, like I mentioned, we conducted an audit with support from Joint Clinical Research Center, and this is part of what is happening. We have what we call the acceptable, non-acceptable, and then risk levels. So where you see very high risk, that is poor. Where you have high risk, that is fair. Moderate, that is average. So if you saw the colors, that means Uganda, we are on average. Low risk, which is good, we are not yet there. And then very low risk, which is very good, that is our desire. The scores you see there were determined to have our checklist scoreable. The audit I mentioned, this is what has happened. We conducted an audit, a safety audit from 52 facilities, and we don't have any under green. We don't have any under low risk. Rather, we have 3 under low risk. We have around 4 under moderate risk. And then high risk, we have 15. Very high risk contribute to the biggest number, which is 57%. The categories of facilities that we visited under the One Health. We had the animal labs and the human labs from national to health center 3. I want to interest you. These are part of our sections from the checklist. But look at specifically laboratory biosecurity. That's where I want us to focus. It is among the poorly scored sections, and it is 23.2%, meaning our biosecurity measures or mitigations are still staggering, and we need to improve. So I want to interest you in this checklist so that we can be able to have it across our facilities. Another performance shows that from the facilities we visited, you realize that we had both government and the private facilities. And because I am government myself, that is what I've managed to score. So I call upon the other different government agencies to do their part to make sure we can improve that score. Otherwise, if I hide my head in the sun and say we are doing so well, then I will be compromising on the standards. So I call upon you to make sure we can improve that score in the next gathering so that we can work with our heads up. Otherwise, that is what is happening. But some of our private labs, which were only 4, they indicated a low risk. You might ask me why only 4 private labs, but the audit exercise targeted those identified only 4 because of the funding gap that we had. Yes, performance of facilities on establishment of occupational safety and health. It is very clear that 55% lacked an occupational health and safety program, which is a backbone for our biosafety and biosecurity, since we are all employed by those different MDS. So we need to think about the occupational health and safety as we do our biotechnology studies. Yes, I want to share with you a way forward as I conclude. The results clearly indicate that we need urgent engagement with our different stakeholders to develop robust biorisk management mitigation strategies. And then we also think that deliberate efforts will proactively identify the potential biosafety, biosecurity risks so that we can be able to avert these identified hazards and threats. Management commitment plays a very big role when it comes to biosafety, biosecurity. So our top managers who are here, you are the biosafety officers of your institutions. So I call upon you to pick interest and make sure the decisions that you take in regards to biosafety, biosecurity are well catered for, and then in that we shall be able to promote a safe and secure work environment. I want to make some recommendations. We've been in existence for the last 7 or so many years, hearing about biosafety, biosecurity, but I want to say that we urgently need a legal framework to be able to design our strategies. Africa CDC is trying to come up with a regional legal framework on biosafety, biosecurity, and we always do benchmarking. So I call upon the different stakeholders here to make sure we can benchmark and have our national legal framework on biosafety, biosecurity, because that's the only way we can operationalize our biotechnology strategies, and then people will be able to embrace them. Training and certification programs. We need to have our professionals certified. So, and I don't know, you've had some universities are now creating training programs for bachelor's degree and master's in biosafety, biosecurity. And I think we have one in the region, that is Tanzania. They're already creating a program there, and they're calling people to go and study biosafety, biosecurity as a profession. The issue of limited funding, that one is on everybody's lips. But what is bringing that is we are doing a lot of siloed implementation. We need to have cohesiveness as partners so that we support the coordination office for biosafety, biosecurity, and be able to expedite our mandate. We have the pathogen economy approach, which is embracing biotechnology. And this is very key if we are to identify our stakeholders clearly. If we know we are going to produce vaccines, we are going to produce our very good organisms, we are going to produce our drugs, then we need to still take you back to the legal framework so that we can be able to do our science when we are well guided. We have national repositories, and I will not mention where they are because for the purpose of being a biosecurity threat. What is their mandate and how are we managing them? What is in lab X or in a repository X? Do we know what they have and its status? So I call upon the different stakeholders here to please build systems to make sure our repositories are well secured to avoid the talk emerging and re-emerging threats, because some of them might come from repositories. I won't feel comfortable if I don't recognize our people. I'll begin with the National Council for Science and Technology for this opportunity to share with you what biosafety, biosecurity program does in the country. CDC that started funding biosafety, biosecurity way back from, I think around 1999, they were funding biosafety. FAO, WHO, IDI, JCRC, NADEC in particular, that we are partnering with, and then Ministry of Health, Ministry of Water and Environment, Wildlife, and then the USAID Most of us here get grants from USAID, and they're all targeting some biosafety, biosecurity component. Thank you for listening to me.
Okay, thank you very much. We need to move on. We are behind time. The next is a colleague from Uganda Virus Research Institute, Dr. Jonathan Cayondo, who is a principal scientist and is heading our Department of Entomology. He's trained, he has a PhD in vector biology and genomics from the University of Notre Dame, USA, and he's doing a lot of research on vectors, bioinformatics, molecular genetics, and is also leading our project on preparing for the genetically modified mosquitoes. So he's going to talk about GM mosquito for malaria control. Jonathan.
Yeah, thank you very much. All protocols observed, and allow me to remove my mask. Yeah. So I'm going to share our ongoing work at UVRI with regard to genetically modified mosquito preparedness. So what we've been doing up to now is laying the biosafety foundation, so that when the time comes to do work involving genetically modified mosquitoes, we have systems and we have facilities and we have the expertise and we have the SOPs that can support that kind of work. Probably you've had a lot of misinformation, miscommunication that GM mosquitoes are here. Maybe I can start right away. They are not. We haven't started that kind of work. We're still far away from that, but still we've been working very hard to make sure we have the right facilities, the right infrastructure, the right teams on the ground to carry out this kind of work. And so that's what I'm mostly going to share today. Also, I want to add that we've had a stall here exhibiting for the last 2 days, and it will continue tomorrow. And that's a good resource to go in case you want to know more about what's going on and where we are at. And we have information and communication materials there that you can feel free to pick, and they'll give you more, because I can't talk more, a lot of that in the 20 minutes that I have. So the structure of the talk will be simple. So I will talk about the malaria problem in Uganda. It will be a background, and then also the mission of Target Malaria in Uganda. And then I'll talk about the laying of the biosafety foundation or groundwork that we've been up to at the UVRI. And then I'll talk about the future plans involving the contained use studies that we'll do, and then the regulatory requirements that go along with that. So malaria remains a big challenge health-wise globally, and lots of people, actually millions, are afflicted worldwide. And most of the problem or the burden is borne most by Africa, especially children, the poor infants and expectant mothers. The economic toll is also enormous, like we know. And unfortunately, progress in the control has stalled, and we've seen rising cases since 2015. And so new tools are needed if we are talking about eradicating it. The case for Uganda in particular is no better. I could just summarize that we're among the 5 most highly afflicted countries when it comes to malaria burden. So we still have a lot of work to do. Now we have interventions that are there. You've heard about nets, you've heard about anti-malarial drugs, you've also heard about probably insecticides, and those are good, and they have saved lots of lives, averting death and morbidity extremely, but they're not adequate by themselves to eliminate, and that's because they're grappling with a number of challenges. If you look at the insecticide, for example, there is resistance that emerges in the mosquito vectors, making them, of course, less effective. If you look at the drugs themselves, again, there's the fear of drug resistance coming, making those therapeutics also less effective. And if you think of things like nets, the mosquitoes are also giving a counter fight so that they evolve and they try to evade the net, they try to bite you when you're not sleeping under the net. So there's an ongoing battle with the humans and the vectors, and so That means the tools that we are throwing at them in our current toolkits are not sufficient to eliminate. So genetic modification can help us combat the disease in novel ways, and that's why it's of interest. The WHO encourages research and exploration of its potential. It's also on the novel technology tool list for APET, and APET is the AU's high-level panel on emerging technologies, and they recommend that genetic modification be seriously considered as a possibility when it comes to integrated vector management, and they think it could be part of a toolkit that complements other tools that are there if it's researched and developed. Actually, in 2017, the high-level panel at the AU and the NIPAD and the Council of Ministers and the Science and Technology there, they recommended that more countries and regions consider that kind of approach in their development and research agendas. And this vision has mostly been taken up by West Africa, where you have the WHO there, the West African Health Organization, WAHO, and also coming together with other players like ECOWAS, and then health regulators, environmental regulators, and ethics experts to kind of see how this could be developed and how it could be integrated in a multidisciplinary integrated vector management, control tool. So they're busy working, trying to look ahead, see how they could harness this kind of technology. So for target malaria, we are a group of experts or a network of, let's say, researchers comprised of more than 200 experts coming from 11 institutions based in Africa, Europe, and the US. And the Uganda Virus Research Institute is one of the collaborating partners in this consortium. So in the malaria endemic countries in Africa, we have teams working in Uganda and in Mali and in Cape Verde and in Burkina Faso and Ghana. And you can see a lot of the activities taking place in West Africa. Now, the mission for Target Malaria is to develop and share a new genetic-based vector tool that can complement other existing interventions in our fight against malaria. So the approach there is to achieve reduction of malaria-transmitting mosquitoes, obviously through genetic modification. And the idea is that if we can reduce the numbers, there's that threshold where you reach that they're so low that you can break the transmission. So that's really the big idea behind this idea. But before you do that, you need to put in place the required infrastructure and the competence and the capacity to carry out that work effectively. And so that's the first phase that the project in Uganda has been carrying up to this point in time. And that's what I'm going to focus on during the rest of this talk. So our research depends on stakeholder engagement and also regulatory compliance. And we have dedicated teams that help facilitate that kind of work. So what capacity have we put up in place over the years? So we have a facility that's built to arthropod containment level 2 standard that we've established back in 2019. And up to now, we've been refining it, trying to make sure it's fit for purpose. So it was designed to comply with ACL2 features, and arthropod containment level 2 has specific features that are characterized by a number of things. Some of those include, for example, limited access to only authorized personnel, and then you have rigorous protocols that are implemented to ensure waste disposal, and you have mosquitoes themselves being reared in species-appropriate containers, and these are being confined in rooms, so that gives a barrier between the mosquitoes and the environment. And then you have additional physical features. For example, the way you enter, you have to go through a set of doors which won't allow you to open unless one is first opened and closed before another one is opened. And then the ceiling and the plumbing for all the piping and also the gaps that are there, it requires to have mosquito-proof netting or appropriate seal. So that's how it's structured, and that's the standard that's recommended by international bodies like the WHO and also the CDC for work that involves, transgenic mosquitoes and those that are not, of course, infectious. And so that's how we built it. So once we built it, we needed to test for its functionality to work at ACL2 as required. Well, because we don't have genetically modified mosquitoes, we had to use a different strain. So we used the strain that we are calling the color variant. So these are natural mosquitoes that show variation naturally. So it's not because of genetic modification. It's like the way you see if you have cattle, some can be black, some can be brown, and some can be white. So that's a natural variation. We call it a polymorphism. So the mosquitoes too, there are traits that you could key in which have genetic bases and which we could use to create particular strains for particular purposes. So for this case, we use that natural polymorphism, which comprised of having a certain pigment at the dorsal side of the thorax and not having it, to distinguish between two strains that we used to try and rear and breed and test the functionality of our facility. Yeah? And we could, of course, sort and sex those strains based on their structures at the pupal stage. So it was not too difficult to do. So the objective for that was to test the capacity of the facility for physical practices and to operate under ACL2 standards. So how good is the containment, and can we implement and run ACL2 standard protocols? And also to test the team's ability to handle genetic crosses, not with GM, but of course with this naturally occurring polymorphism. Because if you are to maintain that trait in the colony, you need to be able to sort and do some crossing so that it doesn't disappear on you. So the team needed to be able to show that and be able to handle that over a number of generations. So in that picture, you see one of the traits where it has a pigment that's white on the thorax, on the dorsal part of the thorax. That's one of the strain. And then the other one is the one that doesn't have that phenotype, and that's the alternative strain. So two strains that we can use with physical features, morphological features, to check our containment. So I said it was creating naturally from local mosquitoes, and then we made the colony, and then we did crossing to maintain that colony, in the laboratory for a number of generations. And so we had to test a number of things. So once, if you do it well, there are certain expectations and certain assumptions that should be or should stay. For example, you should not mess up with the ratios of your pigmented and non-pigmented strain and the sex ratios involved if you do a certain cross in a certain way. So we did the back cross, which should give you a 1-to-1, and each time that's what was observed, and this was replicated over a number of generations, up to 9. So that was good. We also looked at the fecundity, let's call it egg laying or fertility, of the two strains that we isolated, again, because they were the same mosquitoes that we derived them from. They shouldn't be really different from each other. And indeed, the pigmented and the non-pigmented all ended up having the same facility, even when isolated, and continued to be observed individually. So again, we were happy, and that shows that the crossing was done well. We also looked at development times to see how long it takes to move, for example, from egg to adult for each of the strains over many generations. And again, the result was there was no substantial difference between the two, and that's what those graphs are showing, meaning that the team did a good job isolating these two strains, and they are very much able in maintaining the strains and the traits in the colony as expected. And then we look at the environmental rearing controls. Because this is an insectary, you need to rear these mosquitoes carefully. Can we control the environment to specification that the SOPs call for? So there you just see some plots that show the humidity and the temperature are just one of the parameters that we monitored. And again, over time, we could see that in the facility, we're able to maintain the rearing conditions optimal to what the SOPs called for. And so we're happy that we are really very much in control and of the environment in our own facility. And then we also looked at the surroundings, so around the insectarium, what mosquitoes are there, just for us to have a baseline for, in case there was an escape, assuming our color variant escaped, what would they encounter? So we wanted to know what surrounds them. So we observed this through traps over a number of years, and we were able to compile, the distribution of the surrounding mosquitoes that are there. That's a very busy graph, but what it shows is that there's lots of Culex and others, but as far as the Anopheles goes, there were really hardly any Anopheles around our facility. So we know that in the immediate vicinity of our environment, there's really not a lot of Anopheles going on. So in conclusion, this exercise that we did to isolate and maintain a color variant mosquito strain and run it at ACL2, rearing conditions, proved to us that the team was successful. We had the SOPs that were required, and that the facility is really able to contain and operate, at that level. And it was important that we show this before we start to work on, genetically modified mosquitoes. So then, what's planned in the future? Of course, in the future, we plan to start studies on genetically modified strains, and for those, I said the aim is to develop a tool that's modified, that will suppress the natural mosquito malaria transmitting mosquitoes. And to do the suppression, there are two strategies that we're envisaging that could be achieved. One way would be if we modify and target certain genes that will result in biasing the sex ratios of the progeny under the matings that happen. So if that happens, you're having more males at the expense of females. And over time, if that continues, you can see that if there are more male mosquitoes than female mosquitoes, it's the female mosquitoes that spread the disease, but also it's the females that account for the number in the next generation. They're the ones that directly are linked to how many mosquitoes are going to be there in the next generation. So over time, if they're becoming less and less because of that, a time comes when the population becomes low, and then the transmission can be curbed. The other flavor of the same would be we target genes that have to do with actual female fertility in the body of the mosquito. So if you deactivate those, again, you'll have a scenario where the females that are modified are not as fertile because they've been made less fertile. So they're producing fewer offspring. And again, the result is over time, if that trait can spread, then the numbers in the coming generation will be fewer. And so a time reaches where the malaria transmission can be curbed. So that's the big idea behind that kind of strategy. All right? So to do that, of course, there are other things that also needs to be in place. I said regulatory oversight is important. So we are already engaging and been seeking advice from the UNCSD. They're the competent body on these sort of things. And of course, we also have the institutional oversight, the local IBC, and then there's also NEMA. They also have, regulations regarding the environment, and all those will have to be cleared before, of course, we start our work on genetically modified mosquitoes. So that's just a snapshot of where we are at and what we have planned. And I think, that's the end of my talk. And I would like to acknowledge our funders, the Bill and Melinda Gates Foundation, the Open Philanthropy, but then also the government of Uganda, because a lot of salaries of our staff are paid by the government. So it's collaborative work. And then those are the logos for the other participating institutions. So thank you very much.
So thank you very much, Jonathan, for that. Colleagues, we are now going to listen to the presentation we missed in the morning from our colleague who is in Kenya. Is he connected online, ready to give us? So this is Dr. Mugira from the CEO of the National Biosafety Authority of Kenya, who is going to present to us on the current regulatory approaches for gene editing and synthetic biology. All ready? Okay.
To confirm that you can hear me from location in Kampala. You can hear me? Thank you, thank you. I get from the chats that I'm on, and therefore, once again, good morning, colleagues from Nairobi, Kenya, and to you in Kampala, Uganda. We are delighted to be part of this engagement. And I will request that I put off my video to save on bandwidth. I have been having difficulties this morning. I do not know the reason, so that also I'm able to share my screen. Somebody can confirm that you can see my screen? Even by chat, our colleague at UNST. Yes, thank you very much, Dr. Wilberforce and the colleagues across there. Once again, it is a pleasure to join you. I want to say that I truly enjoyed the presentation by your minister at the opening of the sessions on Tuesday, and that was a great eye-opener to what is happening in Uganda in relation to positioning science, technology, and innovation for purposes of driving the socio-economic development of your country. And given that Uganda is part of the East Africa community, what happens in Uganda has a direct impact to what happens in Kenya, in Tanzania, in Rwanda, Burundi, and the rest of the EAC region. So I like to congratulate the people that are driving the science, technology, and innovation agenda in Uganda for the work that you're doing in positioning technology towards delivering service to the people of this region. Having said that, with all protocols observed, I recognize my colleagues from UNST in Uganda. I recall the presentation by your chairperson, Madam Teresa Singova, whom we have worked with a long time, and therefore I send my salutation to her. We have been in this job of preparing a legal framework for biosafety together. So receive greetings therefore from your brothers in Kenya. And with that very simple opening remarks, I like to get into my presentation, in which I am privileged to share the regulatory framework for genome editing in Kenya. And again, as in the screen, my name is Roy Mugira. I recognize that Mugira is a name that is also in Uganda, but I guess Uganda pronounce it as Mugira, and in Rwanda as well. So this is an East African name. I am serving as the acting CEO of Kenya's National Biosafety Authority, the competent authority and the focal point for the Cayondo Protocol here in Kenya. I like to move my slides. Good. As a background, genome editing proceeds in basically 2 or 3 approaches that either may result in an end product that is a classic GMO, or a product that is non-GMO. In certain other procedures, genome editing would start maybe with steps that are GM, and then the final result is not GM. So this is the background upon which the Kenyan regulatory system, the guidelines that I'm going to share, is based on. And we take note that this is what many other jurisdictions, many other countries have taken in terms of guiding their regulatory framework for genome editing. To just touch a little on the genome editing is a newer technology that is targeting the constitutive genomes of organisms without introducing a new gene component, a transgene. and therefore this may not fit properly within the context of genetically modified organisms. And therefore, newer methods of regulation, newer approaches to regulation need to be developed. I am also glad that a colleague who presented a little earlier than before me has also shared the guideline that has been developed by FAO. I think it is in the final stages that is guiding countries on how to approach this. And I think that was a colleague from Global Gene Therapies Initiative. So the approach, therefore, is to recognize that products of genome editing could be either classic GMO, like what is presented in my slide on the extreme left, or that which is not GMO, that can end up in the same way as would have resulted from natural mutagenesis. So this is the approach, the background upon which the Kenya's regulatory framework, or rather the approach towards genome editing, has taken. So the objectives of the guidelines that we published a little earlier in the year is to address genome editing, given that there is no substantive law on genome editing. The guidelines are therefore designed to provide a technical guidance to applicants on the criteria for determining which genome editing organisms or their derived end products are regulated under the Biosafety Act. The Biosafety Act specifically regulates genetically modified organisms, their development, transit, handling, and use. So genome editing, therefore, having those features that I have indicated, may require a kind of special kind of guideline. The key features of Kenya's genome editing guidelines include: one, an early consultation stage, where the applicant fills out a simple form providing information or data that facilitate decision making. So in essence, this is a very, very early stage where we say we have this kind of genome editing process or procedure, we have this product, and therefore it has been developed in this manner. And very quickly, the authority is able to make a decision on whether the product will fall within the category that will be regulated under the Biosafety Act, or it falls outside that regulatory framework. The other feature is an expeditious timeline of about 30 days for decision making at the early consultation stage, and this has been made quite easy to facilitate a quick decision making. Our law allows us to take up to 150 days, between 90 and 150 days in making decisions. But in this particular one, we have allowed for about 30 days quick decision making, and the communication is made to the applicant on the kind of pathway their product or their project will follow. Number 3 is a clear flow of steps on consideration of the various options on the basis of information and data provided. We have presented this in a flowchart that I'm going to show in a short while. And finally, is categorization of genome editing outcomes expected to be regulated under the Biosafety Act. We have provided a table that says this category will follow the Biosafety Act. This other category will be regulated through other regulatory mechanisms. We take note that this is the same approach that has been taken by a number of other countries, including Nigeria, India, and Argentina, and a few others that have also developed this kind of guidelines. And the key feature is the early consultation. Now, we have made decisions on genome editing applications on the basis of the Biosafety Act under the contained use regulation, because these applications are all at research level. So, 10 of these have been approved and are going on within Kenyan research institutions. Number 1, we have some work around the African swine fever vaccine This is a genome editing project that is seeking to develop a vaccine for ASF. Then we have another project on goat for trypanosome resistance by the International Livestock Research Institute. Then we have a project on sorghum to develop striga resistance. Striga is the weed that affects sorghum production and also maize. And then also in sorghum, another project that targets anthracnose resistance. In yam, we have a project that is targeting vitamin A biofortification and also disease resistance. In cassava, we have a project that is targeting nutrition enhancement. And in banana, a project that is targeting nano and colimoviruses and aphids resistance. And again in cassava, a project that is targeting early flowering for purposes of enhancing breeding programs. And another in banana, where the target is fungal and bacterial disease resistance. And finally, potato for potato virus Y resistance. So we used the biosafety regulations and a contained use to approve these projects. They are going on. We expect that they will come up with products that will require to go to the next stage of commercialization, and that is where an early consultation will be conducted to find out of this product that is good for trypanosome resistance. Is it within the classical GMO, or is it within the outside the GMO in the classical sense of genetically modified organisms? And then we shall advise the kind of regulatory framework that will follow. The scope of the guidelines is genome-edited plants, genome-edited animals, and genome-edited microorganisms. As is with the provision with the Cartagena Protocol, these guidelines are not applicable to genome-edited pharmaceuticals for human health use because these are regulated by the Pharmacy and Poisons Board. I recall the presentation by the person from the Global Genome Therapies Initiative that highlighted the potential use of gene technologies in addressing serious medical issues. So this guideline, therefore, may not apply to those because they are regulated under another regulatory provision within the Kenyan legal framework. This is the flowchart for the early consultation on genome editing. It has very simple and direct steps. To start with, the applicant will launch the early consultation form, and then it will be checked for completeness, the usual administrative checks. And the question is there integration of foreign DNA sequence? If the answer is no, then that particular product will be classified as a non-GM, and therefore it will be taken for regulatory systems that are outside the Biosafety Act. So after that, the applicant is notified that the technique or product will not be regulated under the Biosafety Act of 2009, and therefore this falls now within other regulatory systems. If the answer to the question is yes, that there is indeed foreign DNA sequences, then this is taken to be like a GMO, and it is followed by a further question. Is the foreign DNA sequence present in the end product? Like I said earlier, there is a possibility that the process of making the genome-edited product could have intermediary steps that are GMO. But then, what about the final product? Is it GM? If again the end product is as foreign DNA sequences, if the answer is yes, then it is categorized as a GMO, and notification is given so that the regulatory authorities, other agencies can take up the matter, and at the same time, then this is indicated to follow through the Biosafety Act. If the answer to that question is no, again, we go back and classify that as an non-GMO, and therefore it is regulated outside the Biosafety Act. So this is the review, within the table we are showing 15 days, that is supposed to be 30 days. We look through that quickly and we give an answer to the applicant so that the applicant would know what kind of regulatory pathway their product or their technique would follow. So there are two possible decisions on these applications or these early consultation. One, it may be determined by NBA that the genome edited organism or product has no foreign genetic material, and therefore the applicant is informed that this particular process or procedure or product is exempt from regulation as a GMO. Scenario 2 is a determination by NBA that the genome edited organism or product has foreign genetic material. Therefore, the applicant is guided to make a full application that follows a full risk assessment and biosafety approvals in compliance with the Biosafety Act. Now, we have made provisions for the review of guidelines. Remember, science is quickly transforming and changing and advancing, and therefore we have said these guidelines may be revised based on new scientific information. If we become aware of new scientific information that have impact on the approach that we have proposed, then we have made a provision for revising the guidelines. And also, the authority reserves the right to alter its decision if new scientific information previously unknown become available. So this is just to open it up for purposes of revision. Therefore, colleagues, that is what we are doing in Kenya. Thank you very much for the opportunity to share this with you. I look forward to the discussion and any other things that are coming through in this forum. Asanteni sana.
Asante sana. Thank you very much. I think that was very nice, and really to see what our neighbors are doing. It was a very good presentation. We have now, I know we are about an hour behind, but I think I was requested that the biosafety officer from the National Council will give us just a brief on the policy and regulatory matters in emerging biotechnologies. So if the biosafety officer is around, just to give us a brief, and then our discussion will be shortened. It will be very brief, Q&A, questions and answers, and then we go for break. So you can come and give your short remarks.
Thank you very much, Mr. Chairman. The Uganda National Council for Science and Technology, within its mandate, pursuant to the Uganda National Council for Science and Technology Act, has moved along to regulate aspects of modern biotechnology, taking into account the National Biotechnology and Biosafety Policy of 2008. We recognize that these emerging biotechnologies, such as synthetic biology, genome editing, gene drives, are actually expected to transform society, particularly in Africa, where we are expected to leapfrog. However, relevant policy development and regulation for these promising biotechnologies will certainly be a daunting task, as the risks presented will not be fully comprehended or understood until the technologies are further developed through research and development pathways. So, what is our call for African governments? We need to adopt new governance models that seek to manage the dynamics of emerging biotechnology promises, taking into account the risk and also looking at broad constructive enabling governance systems that structure the evolving biotechnology developments in a stepwise manner towards product development. So our question would be, what would be the ideal regulatory model, looking at all those policy and regulatory considerations? We are looking at a science-based, structured, systematically, and data-driven approach. We are looking at starting by systematically identifying plausible pathways, potential harm, and other potential unintended consequences via problem formulation approach or investigative elements. We need to embrace a continuous evaluation and assessment of the social, environmental, legal, and ethical considerations for responsible development of these biotechnologies. I'll take a note on this by highlighting what was published regarding the Chinese scientist. Well, there was an element of crisp babies, which actually was done clandestinely. So this is an element that we need to strengthen as Africa and also Uganda at large. But the good element is that we already have some established frameworks for risk assessment, such as the Cartagena Protocol, World Health Organization, and then FAO. These are resources that will help us move forward. Although at international level, the discussions are still ongoing regarding synthetic biology and also elements related to gene drives and the like. So you will note during the last Conference of Parties meeting, there was an indication to do a horizon scanning and a proposed group to study these deeply to ensure that there is a guidance framework at international level. Members are asking, should we have a separate protocol or we can work within the Cartagena Protocol? And then we need to think about incentives for widespread stakeholder cooperation to produce proactive, flexible governance to inform decisions around the delivery and sustainability of these technologies. So also the regulatory mechanism should be put in place for regular review and updating. At Uganda National Council for Science and Technology, we are working on guidelines on new breeding techniques, and we shall be calling stakeholders soon for input, but the policy proposals are related to issues to do with risk analysis and how we shall manage all those issues. So each technology should be handled on case-by-case basis because each has its unique characteristics. So we implore you to move with us when we call you upon as we deliberate on further policy proposals, but this will also call for reviewing of the attendant policies, especially the National Biotechnology and Biosafety Policy is actually not responsive to the needs of today, taking into account synthetic biology, and also the elements of biosecurity, a need for an extensive consultation related to the policy framework. So that's the current prepositions on policy and regulatory issues for emerging biotechnologies from the Council for Science and Technology perspective. Thank you very much, Chairperson.
Okay, thank you very much. This is the time if you have a question to any of the presenters. We are going to have allow a few of these. We have had, it was a very nice session because it took us through different areas, regulation in the labs, what's happening, the work in HIV, COVID vaccines that we have seen, the work that is being planned in the gene therapy that is quite interesting, exciting as we're moving forward. Genetical preparations for the genetically modified mosquitoes, very interesting. So really we are on the move, and it's very good that National Council is putting us together really to regulate, to ensure that the work we do is moving on very well. We have heard what our neighbors in Kenya are doing, how they are approaching their policies and regulation. So if you have any questions, you'll come. I think we have the microphone. If we can have 2 or 3 so that we don't, one on the other side, there are people who are, yeah, on this side. And then you just mention your name and to whom you are addressing your question, and be brief. Yeah.
Thank you, Chairman. This is Dr. Chibuka from Synthetic Biology Africa Global Catastrophic Biological Risks Initiative. My questions and comments go to Mr. Encordio. Mr. Encordio, thank you so much for the presentation. One thing I wanted to mention or to ask why didn't we have biosafety and biosecurity running together in this? Maybe even the organizers will have to ask, because these two are almost the same things. They complement each other. Then two, I just want to inform members here that we have a project, Synthetic Biology Africa Global Catastrophic Biological Risks Initiative. We have about 4 objectives, but we just started with 2 objectives. One was on creating awareness about global catastrophic biological risks and advocating for legal frameworks. So just recently, 9th to 11th, we had our inaugural workshop where we had the members coming from the Eastern Africa, and it was, I think, a very successful workshop. We shall be engaging many of you here, and after this one, we will be moving to North Africa. So we are moving around the African continent. 3, Mr. Encordio, please, there was a joint external evaluation some time back. Can you kindly tell us how far you have gone with the recommendations of that report? Thank you so much.
Okay, thank you. Let's have maybe 3, and then they will answer those, and then, yeah.
Thank you. Thank you, Chair. I'm Claude Chirumuhuzia, pharmacologist, ethicist. This morning I read 2 articles related to gene editing. One was saying the good, the bad, and the ugly about gene editing. The other one was talking about the dark side of gene editing. The second and third presenter, they gave us beautiful presentations on the positive side, on the good. Can we have some glimpse of the bad and the ugly in relation to gene editing? Thank you.
Okay, thank you. Then we can have those 2. Yeah.
Okay. Thank you very much, Chair. My name is Josephine Naiga, and I just completed my MSc in bioethics. My question goes to Dr. Sise Chityo. Thank you for that enlightening presentation on the opportunities in gene therapy. My question is, how prepared are we as a country or regulatory boards to handle the huge ethical concerns arising from these emerging biotechnologies? Thank you very much.
Thank you. My name is Dr. Mpaji Joseph from Bustama University. I have 2 questions. The first one to Dr. Mujira from Kenya. They seem to have made progress on biosafety and biosecurity. I'm wondering whether measures have been taken to include aspects of biosecurity and biosafety in the training curricula of especially tertiary institutions, that the students as they graduate have a significant appreciation of that aspect. Secondly, to Joseph, my namesake, on biosafety and biosecurity, his presentations seem to have focused largely on containment or management of outbreaks. I'm wondering whether there's an appreciation on the need to prevent the outbreaks, these outbreaks, by sensitizing the communities, especially those communities in areas where there are risks for some of these outbreaks. Thank you.
Thank you very much. My name is Professor Damaina Kanjako from Makerere University, College of Health Sciences. Incidentally, my question is somewhat related to my neighbor, and asking what strategies have been taken or are going to be taken to involve this in training of the young scientists in high school and undergraduate education, even primary school, because these are the innovators of the next generation, and they need to start thinking about these things and how to innovate. If we are going to innovate and produce these are the people we are looking at to bring this out. Thank you.
Okay. Thank you very much. I think let's respond to those. Encordio, I think there are the few. He said, and others also can chip in.
Thank you so much for these wonderful questions and the recommendations. I want to begin with the question on why we don't have biosafety, biosecurity together here. I think it is high time now to, I myself also push it to the National Council of Science and Technology, since we have the biosafety officer here and also the executive secretary in the house, to think ahead of time and review the National Biosafety Forum to have biosafety, biosecurity move under one umbrella, if it is agreeable at that level. Secondly, about JEE. Yes, JEE, the Joint External Evaluation, this is one of our tool that is checking our preparedness in regards to the Global Health Security Agenda Action Package, and specifically on biosafety, biosecurity. What the country has done is we've brought all the MDAs on board to make sure we take them through what the JEE checklist requires, and then they develop improvement projects to make sure we can score better in the subsequent assessments. So the work is in progress. Much as of today, we are still on score 3, which is a basic acceptable score. As a country, we want to be on 5 and above, but currently that is what is hindering us. But if you came and you only measured Ministry of Health facilities, then the score would be different. But we said under national laboratory services, and specifically biosafety, biosecurity, we take on all labs in the different MDAs so that we can move together as a whole. Regarding the prevention strategies, thank you, my namesake. Yeah, I take this as a commendation, and I will engage my team to make sure we can now widen our scope in identifying our stakeholders in regards to biosafety, biosecurity, so that we can be able to include that strategy. Yes, the last, I didn't get her name, but this happened, training of young science in biosafety, biosecurity. I hope that's what you mean. Way back in 2015, when we embraced the Global Health Security Agenda, one associate professor, Majalija from Makerere University, pledged that when I go back, I'm going to change the college and to what it is today. Cover B means biosecurity. So Makerere University is already embracing modules on biosafety, biosecurity, and they're training young scientists. But we want to encourage, I had another government university here, Sitema, and the other private universities, to review your training curriculum to include modules on biosafety, biosecurity, the way you did with quality assurance. I thank you.
Okay. Then we shall have Dr. Mujira also.
Thank you for the questions directed to me. One of them was the bad, the ugly, the good of gene editing. So the title, what I was asked to talk about today in 20 minutes, which was not even sufficient, was the opportunities for gene therapy in Uganda. But certainly, anything in the world has the good and the bad. And we need to look, the same with gene therapy. There are risks, and we have to look at how to mitigate these. And when we look at things that we are adopting, even technology, even when we look at ethics, when we are reviewing protocols for this biotechnology, we have to look, we always balance what is the benefit versus the harm, the harm in what we are doing. And if there are risks or harm, how do we mitigate them? So the risks will always be there. But just to mention a few, there's an ongoing study in the US for gene therapy for sickle cell disease by Bluebird. And this study was halted by FDA. Why? Because 2 of the participants developed blood cancer. And they went to investigate from samples taken even before those participants got into the studies. And they have concluded that these cancers were not due to the gene therapy that was given. So this study has been opened. But these are things and myths, and that's why we need to engage community and tell them, while this may be very good, these are some of the possible risks, like we see for COVID vaccines. The other one that could go wrong, when you edit, you can edit wrong. And when we edit outside of the body or in the body, which is called in vivo. You can edit the wrong area, and you can insert a gene that may stimulate other things. All these things, I think we all need to look at them. The other question was about readiness for these kinds of technology, in particular regulatory. I think we are not ready. But we have the Executive Director of National Council here. And for us, when we are planning our clinical trials, we found that, one, we didn't have policies, we didn't have guidelines in the country to address these technologies. And we've been talking to all the regulatory bodies, and we have been doing also information giving and education. But I think these bodies, they are taking this on head on to put in place guidelines so that we can do what we have to do as researchers. But yes, we are talking to all of them. But they're also here, they can give their comments. Thank you.
Okay, Dr. Mujira.
And I forgot, we have a brochure, again, to educate regulatory bodies and the community. They are out at the National Council of Science and Technology booth. Please pick them. We also have an online one so that we educate ourselves, all of us, as we look for the good, the bad, and the ugly.
Thank you. Thank you very much, Prof, Facilitator. I assume that I'm the one seated on the seat that is vacant next to the colleague who has just spoken. So in Kiswahili, we say, yo ni kiti yangu. So I'm seated there, we assume that, but joining you through this technology. Thank you once again, Prof, Facilitator. There were 2 questions. Number 1, related to one another, measures for biosecurity in curricula for higher learning institutions. And I want to admit, yes, indeed, the Kenya's National Commission for Science, Technology and Innovation is the national focal point for the Biological Weapons Convention in Kenya, and is therefore the focal point. And NACOSTI, as is said in short, is partnered with a project and Denmark, and together with Kenya's Commission for University Education, rolled out a training program for academia, both graduate students and faculty in Kenyan universities outside the formal curriculum. So this has not been infused into the formal curriculum, but there is a robust training program that is going on targeting academia, particularly postgraduate students and faculty. Number 2, we also carry out regular inspection of research facilities and continuous training of faculty to build human resource capacity in biosafety and biosecurity. Going to the lower schools, we are rolling out a reviewed curriculum in Kenya that is targeting to introduce competency-based kind of approaches to curricula. And within this curriculum, there is a reasonable focus on discovery, creativity, collaboration, and cooperation in problem solving. So we are introducing at the very early stage the aspects of discovery, of wonder, of innovation, of creativity in young children at the very early stages in their education programs. Finally, Chair, I'd like to mention a little about the good, the bad, and the ugly, riding very closely on what our colleague from the Global Initiative has put through. I want to indicate that any technology has potential for abuse, and that is why you will be comfortable if you found a military officer in uniform or a police officer in uniform carrying a gun, for example, the AK-47. You won't be worried about that. But if you found some other ragtag people in different kind of clothing and different kind of demeanor carrying a gun, you will run away. And it is the same gun. So it is the same technology in the hands of the wrong people. There is therefore potential for all this technology, genome editing and others, to land in the wrong hands and be used for the wrong intentions. And that is why the global community negotiated the Cartagena Protocol, particularly for GMOs, and also the Biological Weapons Convention, a convention that prohibits the stockpiling of biological and chemical agents. A good example is when the mobile phones were just introduced into our region, there were fears and talk that they will cause ear cancer, they will cause all manner of things. In fact, we went to buy some small gadgets that we were putting on the phone to prevent ourselves from cancer and so on. So any technology has that potential, and that is why genome editing and GMO technologies are highly regulated. It is possible, indeed, for a rogue person to enhance the virulence of a pathogen using genome editing. But again, focusing on the areas that have been presented by our colleague from the Global Initiative, they are very big and huge benefits. Thank you, Chair.
Okay, thank you very much. I think I'm told we are very much behind. I will request that those who have additional questions, you can meet the presenters during coffee time, interact, so that we can exchange more and ask more questions. For me, my final remarks. It's very good, this Biosafety Forum, Biosafety regulations that are coming, but I keep telling my colleagues at UVRI, let's think, because we have had Biosafeties for a long time, even before gene therapy and the rest. Let me hope that in your institutions, as you're talking, we don't only narrow our Biosafety on genes, gene therapy, and modifications. Let me hope you are still strengthening our other safety in the labs beyond Biosafety, because some of these discussions sometimes focus on genes and where we're going. It's interesting, exciting, but safety in the lab and Biosafety is wider. And I hope National Council and the rest take this very much into consideration. Our tradition of Biosafety in the labs is not forgotten, it's strengthened, and we don't concentrate on the genes and gene therapy as we're moving forward. So that's my plea. Thank you very much. Let's put our hands together to thank our presenters for the good and all. So go and have a good, enjoyed break, and we shall be back after. Let me leave the organizers to announce the next items on the agenda. Thank you very much.
Good morning once again. Thank you, Mr. Chair, Professor Chair, for steering us, for steering this session very well and helping us to catch up on time after the late start. And thank you, presenters, for those very informative presentations. The discussions still continue. I think we've formed a network for this forum. We'll ensure that we continue the exchange. We'll go for a short break of 20 minutes. We've lost a lot of time. Actually, one of the presenters in the next session on public engagement from Nigeria has been online and requesting that we give him an opportunity, but it wasn't possible to get him into this program because they were talking about something completely different. So we request that we have a 20 minutes break. The break tea will be at the exhibition area, and we'll be back at 11:20 12:04, and we'll try to catch up in the next session. I'll give a brief introduction of our next chair. The person chairing the Biosafety Communication and Public Engagement session is not Professor Karembu, but we have Mr. Josie Mohanji, a certified PR practitioner who is currently working with Uganda National Council for Science and Technology. Josie has previously worked with the Uganda Wildlife Authority as a PR manager. He has also worked as communications officer at the Economic Policy Research Center. He has worked as a content developer for the East African Community Women in Business platform, and also as a bureau chief with the New Vision. He holds a bachelor's degree in mass communication and a master's in project management. So you'll be in good hands as you discuss issues of biosafety communication. We welcome you all to actively participate in the next session. Thank you very much for your attention. Have a nice break. Thank you, online participants and presenters. We acknowledge your very useful communications, the questions, and the presentations. Presenters and the chair for the previous session, you can pose for a photograph before you go for break as a team on the stairs of the podium. No.